Welcome to the second session of the Catalogue of Life session. We're going to hear a bit about the new extended Catalogue of Life in the beginning and then have a few other talks. You probably know by now, we'll have Zoom chats going that people can ask questions on. We'd like to thank our diamond sponsors that have supported Hadwick and enabled that conference. And let's get started. So I'm going to start myself. It's an easy introduction. So I'm going to show you, thank you, some methods that we use to extend the Catalogue of Life. So what do I mean by extending the Catalogue of Life? There's, as you can see on the right, maybe it's a little bit small, but there's the Catalogue of Life as we know it has two recent species of alligators, for example, in our current version. But there are lots of fossil species existing which are extinct, which we do not track. And the Catalogue of Life is made up of, so far at least, of global sources that cover a group globally, hopefully complete. But fossils are a typical thing that are orthogonal and are lacking in many, many of the groups that we're dealing with. So we try to merge in those missing species that we don't yet have in the Catalogue of Life by using a different source, in this case PaleoBioDB. We do this on the species and genus and family level, but do not really merge any information in the higher classification because that will easily mess up the entire tree. With the exception of some paleo groups and algae, where we're having troubles in the Catalogue of Life because we can't use algae base since a long time any longer, and there's a big gap in algae. So we do a manual classification there as well to give some backbone skeleton, basically, that we can then match other species to. The second thing is we do not only insert new names, but we also do update names in case they are very minimal, the information that we are provided with. So we have especially genera that do not even have an authorship. There's sometimes the publication missing from when it was first published. The type material might be somewhere else, or vernacular names in some languages existing. So we do update existing species as well from other sources. And we track these by what we call secondary sources, because they're not the primary ones that created the species or established the species in the Catalogue of Life, but just gives extra information. So merging new names is really, really highly dependent on matching names. So matching names is the essential piece to get new names in. So if you have here an example of the IUCN Red List, there's some bird names here, and the genus is a homonym. There's two accepted genera, one in the plants and one in the birds. We need to know where to place them, one thing, so which of the two genera, and we also need to figure out which of the names we have already in the list and which we don't. There's some slight orthography changes here, but you can see there's two species that we're apparently missing. And there's all kinds of issues with variations that you have, that people differ, how names are spelled. We avoid any generic fuzzy matches, as you might know from GBIF, which has used that quite a bit, because occurrence names are often far more dirtier than we see in checklists. But we still do some tricks there. Like, most importantly is the gender stemming of the epithet. So people often get the gender wrong, so we do all Latin ending stemmings. There's simple things like ligatures or diacritics, which we expand. There's typical orthographic variations that you see are double letters, T's and L's or something, that are very, very often just typos. And there's often a silent H that we've encountered that is really just the same thing as well. So there's a few cases that we consider still the same name in this matching procedure. And then there's authorships, which are far more diverse that you can encounter. So we're a lot more loose on those. We use some dictionaries for abbreviations or well-known authors, at least, like all those variations of Linnaeus that you can see here. Ex-authors are also a pain. In botany and zoology, the order is reversed, so we're trying different tricks there, and also with the base name and the combination authors, sometimes people just flip them. So there's also some chances there for the matching to go wrong. In general, homonyms are a bigger challenge than people might think. There's like nearly 15,000 genera in Catalogue of Life that exist at least twice, and Erming, which is a really good resource for all kinds of genera that have been described, even lists 33,000 something genera, which is pretty much like 6% of all genera described in Erming. So it's not a rare case that you're dealing with. Just showing a tool in ChecklistBank that allows you to figure out and search for duplicates, or if you like, so all kinds of filters to visualize pairs of names which are alike, which is quite useful. So what can we do to disambiguate those homonyms? There is easy ones like ranks. So biota, for example, is something that we run into. We used it as a Catalogue of Life, has it as the most high-level thing for all life, but there is also a genus named biota, and some matching went wrong in the very early days. There's vertebrata, which is also a subphylum or phylum and a genus. So at least genus group, family group, and higher group names you can disambiguate just by the ranks pretty nicely. Authorship, if existing, is obviously a way to disambiguate. That's really important for species. But with genera, you often have the thing that you just see a species name and you have to somehow place it with this implicit genus that is part of the species, and that obviously does not come with the authorship. It comes with the authorship of the species, but not with the genus itself. But by looking at the authorship, you can also usually detect already the code. So you can see that this is a classic zoological name. It doesn't have a combination author, it has a year, it has no abbreviation. So it is far more likely that this is actually a bird name than a plant name in that case. And then there's the classification. So if the sources have a classification, that's probably a very good source of knowing where it is placed, but they are very diverse, they can be very sparse, and it's really difficult to compare them. So there's two examples that are not as straightforward. So the first one is the Cardus genus in plants, and both classifications have nothing in common if you just look at it. We know by looking at it, it's a plant, it's probably the same thing, but just doing this programmatically is not straightforward. Similar, there's a family down here, which is completely different. It's arthropods, mollusks. It's the same family name though, and they are different ones. So what we ended up doing now is kind of following this human approach that I would think, like, we identify the green stuff, so we're dealing with plants, we're dealing with angiosperms. That matches up pretty nicely, so I think this is probably a match of the genus, while the other ones, we have larger groups, animal, mollusks, arthropods, insects. That is very unlikely that their family is the same thing. And to do that, we created a vocabulary for 44 large taxonomic groups, based basically just on the size of species within the group, that we just took the largest ones, that we can use them to identify those, and then use them to disambiguate or compare classifications in a much more simpler way. That vocabulary is hierarchical, it is non-monophyletic, it's not supposed to be true taxonomy, it's just known groups that somehow can be compared to each other. We've written an analyzer that takes in an entire list of classification, as many strings as you like, and we have a set for each of those entries in the vocabulary. We have an extensive list of higher taxa names that are unique to that group. So it could be family names, order names, or something that we've seen before. but they have to be unique for that group. If it's like in the last example, that family name, that will not be part of those analyzer because it's not unique, right? It's not a distinct thing. The advantage here is that we don't have to treat them as proper synonyms or anything. We can have all kinds of names in there, even non-Latin ones. People do sometimes unplaced something as an indicator of where it's belonging, but so we can put any kind of things that people provide in their classifications in there. And we've seeded them with all the Catalogue of Life names that we're having and quite a few other sources in there as well. But we continually manage that as we find problems with that matching. Nice thing is also, because it's analyzing the whole list in the classification, even if you miss one or two of those names in the classification, it still might figure out where it is based on the tree. It's also very universal, so it applies for all data sets. You can use it as a search filter in ChecklistBank now. And we can provide even those tiny icons that people have some idea of what group they're dealing with. That's the second part of the classification. It's all the animals, which obviously are more than half of the vocabulary. And you can just see on the left side is the number of species in Catalogue of Life. So that's where it's coming from. That is an example of all kinds of bird names that we're having in this analyzer, that whenever we encounter one of these, we know it's a bird. So let me go back to that example that we had before. So we had two Unanthi names that we've inserted from the red list. But if you look more closely, actually in the Catalogue of Life, they did exist, but they were treated as subspecies. So they're different names, but they're all homotypic, and it doesn't make any sense to have the species and the subspecies accepted in the same class, in the same taxonomy. So we need to change that. And yeah, these are linked through a basenym. In this case, actually, there is no basenym. It's all later combinations. And I was a bit surprised to see that hardly none of the data sets in ChecklistBank actually had the basenyms of those. It's only Avi-based that knew about it, but none of them were tracked in any of the sources. There are other homotypic relations also than just the basenym relation. So in theory, you can have replacement names or illegal superfluous names that are all based on the same type. If people provide us with this relation explicitly, we can make use of that to improve the inserted names. But it's impossible to detect that from the name alone, because yeah. So that's then one step we do after we merge names. It's the homotypic consolidation, we call that the species that we've inserted will be made synonyms of those subspecies. We do it this way because the subspecies existed already in Catalogue of Life, and we have basically a list of priorities for the sources that we're merging in, and the higher the priority, that they define basically which one is the accepted one, and all others of the homotypic group will be made synonyms. If we know about replacement names, we can take them into account when we do this consolidation. So we need to identify basenyms though. So some people, we have quite a few lists that give us that relation, but the vast majority is still missing. But you can, as we've seen, or as humans usually do, identify them by the terminal epithet and by looking at the authorship. So we go through family by family at the Catalogue of Life that we've created and compare all the terminal epithets and authorships, group them into what we then think is a potential homotypic group of names, and create that link. There are cases, rare ones, but there are cases where authors really have described within the same family, the same epithet in the same year. So there are such, if we encounter such cases where it's, there's been six original names created by Casey in the Staphylinidae, we back off and say this is something I better do not touch because it seems like they're really separate species, and we don't want to merge them all and synonymize things in this case. that was the introduction. Thanks. There's a few minutes left for questions, or no, not really. Can we maybe start with the next one? It's Diana Hernández, also about extending the Catalogue of Life more on procedures and content level.
Hello everyone. Hola a todos. Greetings from Mexico. My name is Diana Hernández and I am very pleased to participate virtually in this conference and present the progress we have made in the enhanced assembly process of the Catalogue of Life, as well as the challenges we are facing. As most of you probably know already, the Catalogue of Life is the most comprehensive authoritative list of species, having more than 5 million names in total, from which 2.1 million are accepted names of species and a similar number for the synonym names at the species level. It is worth mentioning as well that for several years now, the catalogue has been assembled in the ChecklistBank platform, which also supports the publication and curation of checklists, and it's also the repository for those mobilized and registered through GBIF. It is worth mentioning that the Catalogue of Life and ChecklistBank are recognized as global core data resources. Despite the great contribution from hundreds of taxonomists from around the world, there are still important gaps to fill in the catalog, caused by the lack of experts and communities updating the continuous taxonomic changes and descriptions of new species. For this reason, the Catalogue of Life, together with GBIF, has worked to improve the assemblage process of the catalog, in which we are not only considering the global data sources, but also regional and national sources, taxonomic and management lists, as well as the new species that are continuously being described in different journals. All these new sources are considered as merged sectors in the process, whose information is integrated by filling the existing gaps in the base catalog, so that it doesn't modify the information coming from global sources. The merging process is based upon a series of editorial decisions that prevent integration of errors, as well as the exponential addition of names into the catalog. After several iterations and reviews of the quality content, a new version of the catalog is generated, which we call the extended release, in which the merged names are properly labeled so that any user can be able to identify them. This new version will replace the currently GBIF backbone taxonomy and also will confirm the Catalogue of Life. Selection of additional checklists or sources to be included in the process. As I mentioned before, the assembly process occurs in ChecklistBank, and even though there are more than 50,000 checklists available in the platform, not all of them are included in the process, as we perform a prior filtering of them to ensure a standard level of inputs. In the next talk, Camila Plata will present a more detailed explanation on the type of sources considered in the enhanced process. Here is just presented the mandatory criteria that we consider and some of the content criteria that are considered before adding any source. An important part of the process is to check that the data sources have a low or zero level of errors detected by the ChecklistBank platform, particularly in the name and name usage layers on each dataset. For example, those datasets that have issues in more than 30% of the names, they include do not enter to the process. Another relevant aspect that we consider is the order in which the algorithm includes merged sectors. Therefore, once the data sources have been selected, it is necessary to define the priorities based on the type of list, its taxonomic and geographic scope, if it's a management list, or if it includes nonlinear names, or whether it comes from an automated text mining process. It is important to note that the merging process includes not only scientific names or operational taxonomic units, but also complementary nomenclatural information, references, and vernacular names. All these considerations end up discarding a high percentage of the names available in ChecklistBank. Thus, from the test we have carried out in the recent months, we observed that on average, just over 1% of the names available in ChecklistBank end up being merged into the extended release. In the latest version of the extended release, we merged around 80% more names to the Catalogue of Life. However, this number is constantly changing due to the adjustments we are continually making to the process. If we take a look to the names that are being added by rank, the largest increase is at the family level. In this slide and the next one, we show the species content in each kingdom, distinguishing between accepted names and synonyms. The green numbers represent the base catalog, and the purple ones represent the names merged into the extended release. The numbers of names integrated from multiple datasets digitized by Platzi is indicated in parentheses. Here is shown the numbers for plants, animals, and fungi, and here for bacteria, chromista, and protozoa. In general, the kingdom with the greatest increase in the number of species names in proportion to its content in the base catalog is protozoa. However, the largest number of merged species is in animals. In the next talk, Camila will make a zoom on the merged name of insects. Two important sources of name are Unite and Bold, both of which contribute with names at all ranks, but their major contribution at the species hypothesis mainly in fungi and animal kingdom. We have confirmed that some taxonomic gaps are being filled in the extended release, at least partially. In this case, the base code only has 18 genera of a family and no species. In contrast, the extended release integrated more than twice as many genera and more than half of the species described for the family. This case is a good example that can be assessed by an expert and where specific sources can be searched and added to the process in order to fulfill completely the gaps found in this family. We found that an important number of families and genera are being merged just below the kingdom level. This shows either a gap on the classification on the catalog or a mismatch between the classification in the Catalogue of Life and in the secondary source. We still need to analyze these cases in a greater detail and even with the help and advice of various experts. Spelling variations. The last case that we wanted to share and show still represents a challenge in our process, since detecting them requires a more detailed analysis or user feedback. In this case, a duplicate was generated from a typo in the original publication. The name correctly written was already in the Catalogue of Life, and another name with a typo was merged during the process. This name came from a correctly digitized source. However, it is important not to discard these names from the catalog, but rather to relate them to the correctly spelled name and prevent the error from spreading. In this case, we had to do a specific search in VHL and in the original article and other articles to confirm which name was the correctly written and spelled. We know we still have different challenges in future work that might look overwhelming. And we know we still need to adjust the process due to differences on the content and the nature of information. Some challenges have already been tackled and improved in the process. Others are being discovered through the continuous analysis of the data merged, and others will be discovered through the use of this enriched catalog and user feedback. Thank you very much for your attention. I will be glad to answer any question you have. Muchas gracias.
Thank you, Diana. We have probably just 1 minute. If there's one question, we can answer. But yeah, do it.
Hi, Diana. Thank you for your talk. I wanted to ask, did you try to do automatic reconciliation with BHL? Did you check all the name possible, or you just pick one name? That would be a great improvement.
Yeah, it was just an example to check how deep can we go, but it's not. No, it wouldn't be. It would be impossible to do this kind of search and
well, we
looking for all the
discussed
names. Yeah.
We discussed this option in our group. We'll see how far we can eventually go.
Okay.
Would be nice. Even linking to BHL would be nice already for all names.
Yeah.
But
Yeah.
we have to continue with to the next talk, I guess, where Camila is telling us now a little bit about the sources that we've been using.
Hello everyone, I'm Camila Plata, and today I'm going to talk about the taxonomic data sources from around the world that are helping us to make a better Catalogue of Life. Catalogue of Life is a list of the world's species covering all life on Earth based on a consensus classification and maintained by hundreds of taxonomies. It is used by researchers and by different infrastructures as the main part of their taxonomic backbone. Because of that, we are in a continuous journey of improvement. One of the things we need to improve is to have more infra-specific names, have more synonyms, keep up with the recently described species, not in a yearly cycle, but rather in a monthly cycle, have more fossil taxa, names from molecular registers, and of course, get more taxonomic groups. Until now, we have been working with 164 taxonomic communities that provide information to our infrastructure ChecklistBank. And we add this information in a non-overlapping manner. So for any taxonomic group, we only have one source providing information. So for example, spiders only come from the World Catalog of Spiders, and so on. We gather this information through ChecklistBank, and the information comes in different formats, Darwin Core, CalDP, or even through different transformation pipelines. Nevertheless, to address the gaps that I mentioned just before, we actually need to include even more checklists. And there are a lot of taxonomic communities producing this kind of information. So we want to use regional checklists, management checklists, and other sources. And allow to have a overlapping strategy. That means that now we don't have a one-on-one relationship, but rather have a many-to-one relationship. So many sources can feed information for one taxonomic group. So we have a more enriched product. And this is what we have called an extended release in Catalogue of Life. Luckily for us, we already have very strong communities providing this extra taxonomic information. We are, for example, getting information from GBIF, which is data model has been providing checklists to ChecklistBank for several years now. It also has a very large geographical scope. We also have Platzi, which is quickly digitizing information from literature to the online world, so we can get this information. Once all this information from these communities gets into ChecklistBank, we do a thorough scrutination and data quality process that has been described in previous talks. Now I want to show you some highlights of the sources that we have been merging to Catalogue of Life to have a better product. The taxonomic gaps that are being addressed, but also the geographic context of these sources that we are adding, and I will do some special mentions too. The first type of sources we have are the global ones. We have multi-taxa sources like IDs, WORMS, and ValiDB that we have been using for a long time, but now because we allow overlapping information, we can use them in full and not only for very specific groups. And we also have sources that have a very specific taxonomic scope. These sources are providing more than 700,000 names, which include new names, names that have been enriched with authorships, synonyms, and literature. We also see that these are coordinated by Europe and North America only, so probably these sources have some sort of geographical bias. Nevertheless, we know these sources are doing a great job trying to increase their scope. A very good example of this is Wolf Online, who has 54 consortium members and a strong network of experts that is all around the world. This source is providing the extended release with more than 100,000 names. Many of them are synonyms, which is one of the gaps we wanted to fill. The second type of source is the global management one. These ones are built in essence by national specialists and then gathered by these two initiatives. For the IUCN Red List, we got 21,000 names, many of them also synonyms, and with this we can ensure that Catalogue of Life has the names of any threatened species. From the Global Register of Introduced and Invasive Species, we are getting more than 1,000 names and a really good coverage in terms of countries. With these two sources, we are ensuring that Catalogue of Life has a good coverage of the species that may be of interest for conservation, governments, and policymakers. Then we have the global molecular biodiversity databases. These ones are quite new for Catalogue of Life, but they provide information for many groups that otherwise are difficult to study or are very cryptic, so we wanted to add it to Catalogue of Life. There's also a very specific need for our user, particularly from GBIF, to have the information of the occurrences very well organized. So we don't want species hypotheses or beings floating around. We wanted to keep them inside of a backbone on a very well structured hierarchy. So they are also important to add them to Catalogue of Life. Here we also have very interesting results. Unite provides more than 2 million names. Most of these are species hypotheses, but they are covering taxonomic gaps that we have. For example, in fungi is adding many names. We know fungi can be cryptic, difficult to describe, so we are getting this information through Unite and also through species hypothesis. And an unexpected finding is that we also get information from green algae. We really like good sources for algae in Catalogue of Life, so we are closing this gap with Unite. And very similarly with Bold System, we are getting information from insects. This is also another group where we have gaps, and from red algae. So unexpectedly, the algae gap is being closed by these molecular biodiversity databases. Then it comes the regional sources where we have national checklists, endemism checklists, catalog of type of species, or conservation areas. And with these ones, we wanted to really go deep into covering geographical gaps in the global south. What we have found is that we could use 140 checklists from around the world. We found information from all the continents and also from the biodiversity hotspot areas like Africa, Asia, Latin America, and the Caribbean. Nevertheless, we found that Europe is still producing a lot of checklists, which is really good, and we were able to add more names from these regions, but still the southern regions is still not that well covered. So we need to find a way to discover more of these national and regional lists to keep on closing the geographical gap. Then we have sources like Platzi and Biodiversity Data Journal that are allowing us to have very quickly more information. And here I'm going to focus on Platzi because Platzi is providing 20,000 lists. There are lists that sometimes provide 1 or 2 names, but this information adds up and is helping to close all the areas of gaps that I was showing before. So it's providing now 9,000 infraspecific names, 20,000 synonyms of species names, also adding 8,000 recently described species. These are species between 2020 and 2023. Probably these species at some points will be provided by the global sources that we usually use. Nevertheless, with this, we have a shorter gap of time between the species being described and arriving to Catalogue of Life. We also have some potential fossil information there. We're still working on that one. And we also are covering other taxonomic groups that are complicated, like Coleoptera and Hymenoptera, where we don't have a specialist for all the families. Then for the geographical analysis of Platzi, we see that around half of the checklists that are being provided are from a specific country or a specific region. And very interestingly, is Asia, Latin America, the Caribbean, and Africa, the regions that have more checklists being added and more names being added. So then, together with the world, the managements, and the molecular registers plus Platzi, then we can actually get a good geographical coverage. Of course, there's still a lot of work to be taken part to really don't have gaps, but we see a progress with this combination of sources. And here I want to close with an invitation for you to help us to build a better Catalogue of Life. Please publish your checklist or recommend us on checklists to keep closing these gaps, both the taxonomic gaps and the geographical gaps. Any regional list that you may have can help us make a better Catalogue of Life. Please be mindful about the licensing. Use a CC BY license. There are very good resources out there that we haven't been able to use them because they are not licensing properly. Also, keep in mind the data quality, so we can usually merge and keep the Catalogue of Life very tidy. You can also report issues to our GitHub account. So anything that you see wrong, anything you see missing, you can let us know and we can quickly act to improve it. Thank you for your time and enjoy the Congress.
Thank you, Camila. We have 1-2 minutes for questions. Yeah.
Camila, wonderful talk. I have one question. So let's say I supply a checklist to Catalogue of Life. You may add some names from Plaza or presumably from some other sources. Is there an easy way to filter out and see those names which was added to provided checklist?
Yes, we have like an indication mark on ChecklistBank that says what has been merged and from which source. So you can quickly filter out what has been merged.
Maybe I can add that you can search by a higher group. So you can use your group and then only list results which come from merged sources. So you can see just the species that have been added to the group. We will be working on something to provide for the GSDs in the Catalogue of Life, the global sources.
That would be useful.
We really want some page where you can go to and see like these are names that we have added in the extended release and which are not present in use, and that we can have some feedback interaction over time.
Yeah, that would be great.
But yeah, that's coming at some point. But.
There's a question.
Is there one more? Yeah.
So we have question from Stephen Garnett, which says many countries have list of species that are protected, listed in their legislation, many of which are not digitized. To what extent have you been able to add these lists to ChecklistBank?
Right now, we haven't made like an active effort for this list to be published. We will have to just try to invite more people to do so. What we have done until now is to try to find them, and most of them have like very close license, not that close, but they are not commercial licenses. So we have been approaching them with the help of GBIF and asking them to change it to CC BY. So that's what we have been done until now.
This from the European Environmental Agency, those lists, I think there's 40 or something, I know, Berne Convention, and these lists have been added already. We gotta skip on to the next talk. Sorry, it is Lauren Russ from Colombia also, who's telling us about the global land plant taxonomic resources.
Hi, my name is Lauren Raz, and I'm coming to you from Bogota, Colombia. I'm one of the co-chairs of the World Flora Online Council, together with my co-author, Mark Watson, from the Royal Botanic Garden, Edinburgh. The title of our talk is Towards a Global Land Plant Taxonomic Resource for Science and Society. That resource, of course, is World Flora Online. WFO is an online portal with up-to-date scientific information made available on all known land plants using FAIR principles. But WFO is also a community made of member institutions and participating plant taxonomists. WFO derives its mandate from the Global Strategy for Plant Conservation, approved by the parties to the Convention on Biological Diversity. Target 1 of the GSPC was to produce an online flora of all known plant species by the year 2020. But WFO aims to support plant conservation and sustainable development well beyond 2020. WFO was recently recognized in the Kunming Montreal Global Biodiversity Framework of the CBD. The framework includes complementary action number 21D to promote the continuous updating of the World Flora Online. WFO also recently produced a strategic plan taking us up to 2030, so this is a long-term commitment for us. WFO replaces the plant list, which was the fulfillment of Target 1 of the previous Global Strategy for Plant Conservation. Some of you may be familiar with this resource, but it's been inactive since September 2013. The current WFO portal has 3 core elements: the WFO plant list, species pages, which include descriptive content, references, maps, and images, and the About pages, which provide general information about WFO. The WFO portal launched in 2017 and was revamped in 2021. It currently has 1.6 million names, including over 377,000 accepted names, over 400,000 distributions, 164,000 descriptions, 1.5 million references, and over 56,000 images. The About pages and WFO plant list components were launched in 2021. The species pages compile the descriptive information, and the information that's fed into the species pages is provided by our consortium members. This brings us to the community aspect of WFO. We're an international consortium of 52 institutions and growing. Members need to sign a memorandum of understanding, but there's no cost to join. We have consortium members located on all continents, but we are doing continuous outreach to increase membership. But the heart of WFO is really our taxonomic expert networks, our TENs. These are groups of specialists who are working towards a consensus classification for the WFO. Ideal TENs are open, inclusive, representative, and collaborative. Generally, they work at the family level, although we do have some what we call high-level TENs, such as bryophyte, pteridophyte, and conifer TENs. Also some TENs working at the order level, like the Caryophyllales TEN, and other groups that are working below the family level on tribes or subfamilies. Some of our TENs predate the existence of World Flora Online, but many of them are new, spurred on by WFO. And we recently did a recruitment drive at the International Botanical Congress, and I hope that from that effort we'll be seeing a flood of new TENs in the coming months and years. We currently have 49 TENs curating the taxonomic backbone. This covers about 54% of the families in the WFO backbone and translates to 45% of the accepted species that are covered by TENs, and they're doing a great job. They've already curated about 38% of the accepted species. We expect to bring on 7 new TENs within the coming months, and that's not including all of the expected TENs from our recent recruitment effort at the International Botanical Congress. So over the last few years, we've seen tremendous momentum in WFO, and this has to do with recruitment of TENs and the involvement of the taxonomic community. There's also a really important technological component. Over the last couple of years, we've developed a new tool called Racus, which is an editorial platform for managing the WFO taxonomic backbone. This was created and is currently managed by staff at the Royal Botanic Garden, Edinburgh, and access is provided to the TENs, many of whom are able to edit their data directly in Racus. Racus can also ingest data from other platforms, such as Edit, TaxonWorks, etc. So Racus is browser-based, has user authentication via the ORCID ID of our participating taxonomists, and users are assigned to the specific taxonomic group that they work on. So the taxonomic expert networks edit directly in Racus, or they can feed their data to our TEN manager, who is the coordinator of the taxonomic expert network activity. The TEN manager is also responsible for making updates directly in Racus and is responsible for tasks like data cleanup, deduplication, and other database administrative tasks. So there's constant coordination, communication between the TENs and the TEN manager. The TEN manager is also responsible for coordinating 2 yearly data releases, which happen on the solstice, and the WFO plant list is the product of this data release, which is made available via the World Flora Online portal and also via external sites like Zenodo, ChecklistBank, etc. So while the TENs are absolutely key for maintaining the taxonomic backbone up to date, our TEN manager also plays an extremely important role in this. He has additional workflows, including monthly imports from IPNI, the International Plant Names Index, through which we bring in new names. These new names are fed to the TENs, who make corrections, and these corrections in turn are fed back to IPNI. We also import our default classification from the World Checklist of Vascular Plants, which is produced by the Royal Botanic Gardens Kew. This is a source of additional names and updates to the classification, and in many cases our TENs use the WCVP data as a baseline from which to begin their editorial work. In addition to IPNI and WCVP, we've also begun importing information from Platzi, which we expect will help the TENs in their work while growing our literature database. So, putting it all together, the TEN manager and the TENs are continually updating the taxonomic backbone in Racus. This backbone is used to attach all of the descriptive content in the WFO portal to create the species pages. Twice a year, the WFO plant list is also generated from Markus via a WFO plant list API and is made available on the WFO portal and is also shared with Zenodo, GBIF, and the Catalogue of Life. And the API is also available for anyone to use anytime, anyone who's interested in connecting the WFO plant list to their own applications. By making the WFO plant list available through so many channels, we're also increasing the visibility of our TENs and their hard work, and hopefully providing incentives for new TENs to join. With the innovation of Markus and greater involvement of the TENs, this combination has been a game changer for WFO. Although the total number of accepted names has remained fairly stable, there's been tremendous burst of activity. 142,000 more synonyms have been added only in the last 2 years, close to 1 million in total, and 34,000 fewer unplaced names are now in the database. In just the last 6 months, we added 38,000 names and edited almost 1 million records. So while we've made major strides in the last 2 years, there are still some significant gaps in our taxonomic coverage. 70 families with over 500 species still lack a 10. But with the recent recruitment efforts and all of the enthusiasm that we saw at the International Botanical Congress, we're feeling very optimistic about the future of WFO, and we're very much looking forward to continuing to deepen our ties with Catalogue of Life and help bring high-quality plant information to an ever wider public. Thank you to our wonderful 10 manager, Alan Elliott, to symposium organizers Olaf Banke, Joe Miller, and Don Holborn, to our hardworking taxonomic expert networks, to the conference organizers, to our home institutions for supporting our participation, and thank you all for your attention.
Thank you, Lauren. If there are questions, a few minutes are left. Okay.
So hi, thank you for this interesting talk. My name is Lien Reestroeven from INBO, and I was just wondering, this Markus platform you're talking about, this sounds like a very good platform, because we are also thinking about a similar initiative for updating local GRIS checklists. So I was wondering if the platform, if we would like to develop such a thing, similar thing, who should I speak to, or is the source code open available, or yeah, can you give me some guidance about that?
Yes, so the platform is being developed at the Royal Botanic Gardens, Edinburgh, and the main developer, I don't know if he's in person at Chadwick, is Roger Hyam, and I'm so sorry I forgot to include him in the acknowledgements, but he needs to be acknowledged because he's done a fantastic job. If you want to get in touch with me, I can put you, I can share my contact information, or you can write a message through the World Flora Online website. And as far as I'm aware, all of the code is in GitHub, so it should be available.
Okay, thank you very much.
Yeah.
Hi, this is Rod Page. As a zoologist, I find plants difficult to understand because I think you have 4 global plant lists. Something like that order. There's WFO, there's a list in Leipzig, Q has at least 1 or 2. Why are there 4? And which one does Catalogue of Life use, and why does it use that one? As far as I'm aware, it doesn't use WFO. So what's going on with plants?
Well, there's good news on that front. We're moving towards greater integration. So there have been kind of these independent initiatives that have been going on. I would say the general trend in our community is to have a community-led list where that's the most inclusive, possible, which would be at this point WFO. The different lists have different histories. in some cases, they're individual efforts by single people that don't have a very broad horizon in terms of their sustainability. I would say that our model and our, strategic planning has greater sustainability built in. So I think you're going to see in a very short time convergence on WFO.
I'd like just to, I can agree with all of that, but I'd just like to add that Catalogue of Life is using, so there's Michael Hassler's list, which Catalogue of Life is using for more than 50% of plant names at this stage. WCVP is the Q global one, which Catalogue of Life uses for most of the remaining. And WFO is rather new for the Catalogue of Life. We use it now for the merging, but we gradually will be replacing things, and hopefully we all agree that we will merge to this World Flora Online being the main source, but that is a bit political, obviously, also. But like Lauren said, the more openness, the far wider base of people involved, I think that's the way to go, and not have like one-man shows. Even Leipzig Catalogue is just one person really that does it, and that's not very sustainable. So our next talk is by Cecilie from GBIF.
Oh, tall. Sorry. No, it's okay. I can do that. Okay. Hi, everyone. So I'm here to just please ignore this very long title. I'm just here to talk about DNA in GBIF and Catalogue of Life. That's the basic of it. So what is DNA derived data? It basically means, I want to talk about this, it's data that's coming from two different sample types, and then there's, of course, all the processing methods, but just let's go into the sample types first. So, there's environmental samples. This is DNA coming from not a specific organism, but from the environment around that organism, or multiple organisms. So you take a soil sample, and then you get DNA from that. That's the short presentation. And then we have single organisms. This could be voucher specimens in museums where you use, the taxa you are extracting DNA from, and you use this as a reference. So you can include it in a reference database. So basically you can detect all the species you find in the environmental samples based on these references coming from single organisms. So how are these reference databases generated? This is important because you already heard about bins and species hypotheses. What they are basically multiple sequences that are combined into one species hypothesis that's unite and that uses that name, but you also have the Barcode of Life who use bins. It's all the same thing, really. It's an algorithm that combines all these various submitted sequences into a hypothesis that they are the same thing, they're the same name, but there's also a list of, this is species A, we believe this is species A because if you have 100 sequences, maybe 85 of them were reported to be the species, but you can also have some of those sequences reported to be under different names. So it's constantly changing. The algorithm are constantly making new hypotheses on what do these sequences represent, which name do they represent. So in DNA barcoding, as I said, it's one organism, it's one record, it's one reference. So you take DNA, you process it in the lab, you clean it, and then you have a record. So I'm going to talk about this in a GBIF context, that's why I say GBIF record, but it could be anywhere. In metabarcoding, where you have an environmental sample, you have multiple species coming from one sample. The bioinformatics pipeline, the data cleaning part, is a bit more complex. I should also say you have shorter reads. You have, maybe people will get angry. You have less evidence that this is actually the species, where in the barcode reference, you usually have longer reads, which basically just means you have more information. And then the shorter reads are then usually shared as multiple records within one data set. There's also a higher risk of false positives and negatives with metabarcoding, but it's a really useful tool to examine species distributions, which I will get into later. So, for example, it's a very valuable tool to reveal the presence of less charismatic species. I'm going to show you a use case here. So we have a sequence. It's assigned to this scientific name I forgot to mention before in Catalogue of Life in GBIF. A scientific name is basically the bin or the species hypothesis. It's not the Linnaean name. It can be associated with a Linnaean name, as I show here. So in this case, it's most likely that this bin is this little fly. And this little fly is a actually quite well-referenced fly. And you can see from this GBIF occurrence map, all the single dots are coming from museum collections over the last 15 years. It's references that are generated. And the cluster you see is from one research project for the month of June of 2018 and 19, where citizen scientists sampled random samples of insects in basically using big nets on top of on the roof of cars. And then they had, we sequenced them all, which gave a complete distribution of this species across Denmark. So we can reveal with one single project with metabarcoding what 15 years of references supporting this, this work. And this is probably a very common species in Europe and beyond, but we won't see that unless we have DNA barcodes, because people are not collecting this fly, because it's boring. Anyway, DNA-derived data in GBIF. It started back in 2014, where we had the first data set. Back then, it was published as Linnaean names, so we didn't have bold bins or species hypothesis. But in 2018, by collaboration with the Unite community, we were able to include this, and we had stable identifiers for ITS, which is species hypothesis. And then thing escalated with the pipeline, automated pipeline set up with the ENA, the European Nucleotide Archive, and we also had the backbone to support bins and species hypothesis as scientific names. And then in 2021, we had a DNA-derived publishing guide set up, so we could enable publishers to publish their DNA data. So I'm just going to talk a bit about how it works. Again, we have multiple sequences that can be assigned to multiple scientific names. This is an example again from the bold checklist in GBIF, which can be associated with Linnaean names, which then shows up on gbif.org. As you may saw on Camila and Diana's slides, the bins are basically treated as unranked taxon ranks, so they appear under the Linnaean name, but as an unranked scientific name. One thing to be mindful of is that multiple bins or species hypothesis, this is species hypothesis, can be associated with the same organism. This is because of the reference databases, so what we hope to see with increased or improved taxonomic databases, we will see more separation into more species, but it's something to be mindful of. But the focus of this talk is, why does it matter to switch the GBIF backbone to the extended Catalogue of Life? So going back to that, GBIF backbone usually updates 1 or 2 times a year. It's quite a manual process. And you know, many of these reference databases are updated monthly, and it really matters. I have an example from back in a couple of years back. Germany made a really big effort to do references on Diptera, and just by publishing once, they improved the taxonomic coverage of Diptera by 25%. So you can see that if that data is actually in GBIF, then suddenly you increase the searchability by far for many of these flies. So what we hope to see when we switch to Catalogue of Life or the extended release is that we will have a much more frequent update of the taxonomy for DNA and other things, of course, maybe more in track or in line with what the reference databases are currently updating. So some of the potential changes we envision for GBIF and by switching to Catalogue of Life is we hope this will improve our coverage of reference databases. We have Unite, we have Barcode of Life, we have more, and it's slowly increasing. But one thing we really hope to see is whether we can be able to re-annotate records if sequences are shared. So we don't want to overwrite what the publishers gave us, for sure, but we also in a reality where taxonomy for DNA becomes quite quickly outdated. So, and the chances that a publisher are able to update their data set by a monthly basis are probably not quite likely. So what we hope to be able to do is that we can re-annotate them with the newest taxonomy, so that's also will be possible for users to search, which will increase the discoverability and linking of related records as well. So just as an example, this is one data set where we have re-annotated over time. We started back, you can't see that, but back in 2019 until, I think 10 days ago, where we have been re-annotating, and as you can see, it really matters. You see an increase in the amount of matched records. It's how well they're matched. So the dark line, the dark bar are the exact matches. So those are maybe the most interesting. And then you have a decreasing confidence in the matches. But overall, we see a trend that you can get a better match and better naming of your sequences if we do re-annotation. So with that, I want to stop. Thank you. We also have a DNA at GBIF if you have any questions, dna@gbif.org email. So you can also write to that, but you're welcome to contact me as well. Thank you.
Time for one, two questions online? Yeah. No. 2 minutes, yeah. Any questions, or is there anything online? No. Well, then we go on to the next and last talk by Sherrin from Australia.
I would like to start by acknowledging the traditional owners of the land on which I live and work, the Wurundjeri people of the Kulin Nation, and pay my respects to their elders past and present. I would also like to recognise their continuing connection and custodianship of the lands, seas and waters. Today I'm going to talk about our experience of using ChecklistBank to build a composite taxonomy for a large biodiversity aggregator. I'll start with a brief introduction to the Atlas of Living Australia, the ALA, for those of you who are unfamiliar. The Atlas is Australia's national biodiversity infrastructure. We aggregate over 145 million records from a variety of data sources. We have over 820 data partnerships, and various components of the ALA's infrastructure are in use in 27 countries. We are also the Australian infrastructure node for a number of international projects, including the Biodiversity Heritage Library, iNaturalist, and GBIF. We aggregate a wide range of biodiversity information, preserved specimens, living specimens, and human observations from a variety of sources, from natural history collections, science agencies, conservation groups, individuals, and Indigenous ecological knowledge holders. That aggregated data is used for scientific research and decision-making, government planning, policy and assessments, education, citizen science, and more. But to use those 145 million occurrence records effectively, they need to be put in the right spot. The key components of putting things in the right spot are the ALA's taxonomic backbone and its name matching service. Today, I'm going to talk about the taxonomic backbone, how we put that together, and how we use ChecklistBank to review and improve our most recent update. I'll also talk about some additions to ChecklistBank that would help us in constructing our taxonomy. We describe our taxonomic backbone as a robust and comprehensive taxonomy. But what do we actually mean by that? The species names in the occurrence records that we're aggregating have come from a diverse range of taxonomic checklists, both current and historical. In building our taxonomic backbone, we're not aiming to be a source of taxonomic truth. Rather, we're aiming to achieve a specific outcome. And that outcome is to be able to assign every occurrence record to the correct taxon. That means having the best available name for all the data that's coming into the ALA. We've been working on changes to how the taxonomic backbone is constructed in order to improve that name matching. To construct the backbone, we take data from authoritative taxonomic sources such as the Australian National Species List, the New Zealand Organism Register, and the Catalogue of Life. We also use information from non-taxonomic sources such as state and federal conservation, biosecurity, sensitive, and migratory species lists. These lists have overlap in terms of names, but their importance is in filling the gaps. All these sources are pre-processed to create individual Darwin Core archives. Our taxonomic compiler takes those Darwin Core archives and combines them to produce the taxonomic backbone. Different sources get different priorities, which determines which source gets used where there is overlap. This latest build of the taxonomy was the first time that Cam and I had worked together on this process. And while we were using the existing approach to building the taxonomic backbone, we were also testing and experimenting with ideas about how the process could be improved and refined. The issues that we needed to tackle fell into two main groups: things we needed to fix in our process and things we needed to work on with external agencies to fix. One other issue that we had was all the data processing was done locally on my computer here in Melbourne, while Cam works in Canberra, about 665 kilometres away. ChecklistBank became an extremely useful tool in our collaborative process by allowing us to easily share and review various stages of our taxonomic construction process. We use ChecklistBank at two different stages of the build process. Firstly, we used it to review the results of the pre-processing applied to our data sources. This enabled us to see that we'd removed or modified only the branches that we wanted to, and to easily examine the taxonomic tree via the browse functionality. Once we'd completed a build of the taxonomic backbone, we uploaded that and investigated issues using the reporting tools. Once again, being able to easily browse the taxonomic tree was of enormous benefit. The ability to see the difference between those uploads allowed us to confirm that things that we'd done to fix a particular issue had worked and hadn't introduced new issues. And when we had our final build, we made that private checklist available to reviewers within the ALA and outside the ALA. While ChecklistBank has been an extremely useful tool for us, there are a number of things that would make our lives even easier. We've split our wish list into two parts, based on how technically challenging we think those requests are. The first item on our wish list is about the display of scientific names. We get a lot of informal names coming through the conservation lists, and the name parser gets a little confused. When browsing the taxonomic tree, we see a lot of apparently duplicated entries. For example, a series of names that are just Eucalyptus sp. it would be great to have the option to just display the scientific name as provided in the Darwin Core Archive. Also, because our taxonomic backbone combines a number of datasets, in addition to filtering by taxon rank and status, it would be great to be able to filter by the data source ID. That would allow us to easily compile issues that need to be discussed with individual data providers. Finally, when we've created a filtered list of names, we would like to be able to download that list for offline review. So on to the more possibly technically complicated items on our wish list. The first is the ability to compare two private checklists from different users. We're encouraging our data providers to make use of ChecklistBank, and it would be great to compare our version of their checklist to ones that they've uploaded themselves. The second is the ability to edit a checklist within ChecklistBank. At the moment, we're managing the additional names that are coming in from conservation lists and sensitive species lists via a series of spreadsheets. Just due to the nature of spreadsheets, mistakes can be introduced where a name gets misspelled or a synonym is assigned to the wrong accepted name. Being able to add species directly into the appropriate place in a checklist or make edits to names would make the entire process that much easier to manage. It would be even better if those changes could be tracked to both document the changes, but also potentially using them to check for those changes when a new version of the checklist is uploaded. In a similar vein, we would like to be able to upload a checklist into a specific point or points within an existing checklist, rather than overwriting the existing checklist. Once again, using the conservation lists as an example, we could upload those species into an existing checklist and discover if they will all slot into the hierarchy that exists in that existing checklist or whether we need to make changes. We may be approaching ChecklistBank from a different direction to a lot of users, but it never hurts to ask. Once again, we've found ChecklistBank to be extremely useful in our pursuit to build a better taxonomic backbone, and look forward to working with you in the future. Thank you. And if the internet has been kind and I've managed to join you virtually, I'm happy to answer any questions.
Thank you, Simon. If you can put up Simon already, I think he has something, he wanted to say something, if I remember correctly.
Good morning. Can you hear me?
Very well, Simon.
Yeah, you can hear me? Great. First off, I've got to make an apology that having done the slides and recorded that presentation about 12 times, I failed to notice that I'd left off Tanya Lady from the title screen. So my sincere apologies to Tanya for leaving her name off. It's that thing you look at the morning before and it's done. Happy to answer any questions if there are. Apparently.
Are there any questions online? Yeah.
Sorry, Arthur. I've got a question. Why do you use New Zealand Organisms Register rather than the, well, as I understand it, and I could be mistaken, more up-to-date Biota NZ dataset?
Siobhan, is that you?
Yes, that is me. Sorry, I should have introduced myself.
Oh, no, it's fine. It's fine. Look, that's something that we've been traditionally using, and we are looking at currently updating those. So we only use the plants from the New Zealand organism register because we are building our backbone not just for the ALA, but it's also used for the virtual herbarium, which includes New Zealand herbaria. So we are looking at that. We are looking at updating, getting different sources, or a more recent build of Enzor and discussing that sort of perspective. But yes, we're doing it for the plants.
Yeah, okay. Thank you for that.
Section.
I
No
have
worries.
actually I have actually asked on Twitter, but I'll be getting to them personally, why the Biota NZ isn't shared on ChecklistBank as far as I'm aware. I can't see it. So I'd quite like it shared, obviously. Yeah. Okay, thank you for your answer.
Oh, thanks, Siobhan. It's strange not being able to see everyone in the room, and I'm sort of sitting here.
Right. So I think there are no more questions around.
Great.
Right. Yeah. Thank you, Simon.
Cool.
Yeah.
Thanks, Markus.
That's the end of the session then. Thank you. Thank you for coming around.